Good morning, good afternoon, and good evening. It's a pleasure to present the work that we are doing on behalf of all my collaborators. We are thankful for the BRAIN Initiative for their funding, as well as for the invitation to present this. Our work is on ultra-high resolution brain PET scanner for in vivo autoradiography imaging, and we call it the SAVANT system. Before I start, I would like to start with a slide of history, just to point out that the very first PET scanner that was ever invented was actually for brain imaging. So this first device was invented here at Mass General Hospital by Gordon Brownell in the center that was the predecessor of ours. And it was used by Bill Sweet, who was a neurosurgeon, to delineate areas of surgery intervention. So from the very first beginning, PET and brain were associated together in terms of the research and the applications. So the rationale for our work and why are we doing this is that although we have come a long way in terms of performance on the PET side of imaging with high sensitivity now in the picomolar, it's the highest of any imaging modality. Compared, for example, to MR in vivo, we are at 10 minus 12, so picomolar sensitivity instead of nanomolar, so 10 orders of magnitude in terms of sensitivity gain. We can image hundreds of thousands of cells, and we can quantify that. The main limitation is spatial resolution. If you think about a clinical scanner, about a 4 to 5 millimeter spatial resolution, that amounts to about 100 millimeter cubed spatial resolution cubic, compared to a 1 millimeter cube with spatial resolution in MR. So we are far from the spatial resolution needed for imaging the brain accurately with the current spatial resolution. And that's really where the motivation is for the work we're doing. And we would like to come to no less that to 1.5 millimeter cube to become very comparable in terms of spatial resolution to MR. And while having the very high sensitivity and, of course, the anatomic information. And there are several concordant events that push for this and need this kind of resolution currently. The first comes from new developments in terms of neurofibrillary tangle tau, the helical perifilament that are binding in the brain intracellularly, where there has been several tracers that are PET tracers, radio tracers, that map these. The very first was for tau-CPR, Theta-7, and then since there have been second generation compounds. And we have been lucky to be one of the first groups that developed all the kinetic modeling for these first two tracers that have shown clearly that these tracers are literally mapping what you would see on immunohistochemistry, except that you could do that in vivo in the living human. This is the autoradiography of brain slices that shows exact distribution similar to the immunohistochemistry. But as you start looking at tau deposition in the normal aging brain, programmably before any memory complaint, you can see, and we have shown this in 2013 as recently as yesterday in a paper that just appeared in STM, that there's one of the very early areas where everything happens is in the transentorhinal and trianal cortex. And as patients, as human subjects age, before they become patients and before they become mild cognitively impaired, that's one of the very first areas where we start seeing accumulation. The problem with this area, the intrarenal and transentorhinal, is they're less than 1 cc in volume. So well below the spatial resolution of PET to have quantitative assessment. You may be able to see them, but you can't quantify them. Another small structure that is no less important is the Locus Ceruleus, which is literally a blue dot. It's a blue spot. This is where we produce most of our norepinephrine in the brain. And this site has widespread projections to the entire cerebral cortex. It's the primary seat of neuroadrenergic neurons. And the loss of norepinephrine is linked to many, many cascade events in neuroinflammation, but also in neurodegeneration, in Alzheimer, Parkinson, MS, etc. So we would really like to be able to see and quantify the structure, which is 2.5 millimeter by 2 millimeter. And again, the challenge is that on PET, this is what it looks like. It's like a wing-shaped appearance from our colleague in our center, turn single. And there's no way you can quantify that easily today with PET because of the resolution. And the third component of where we need this high spatial resolution in our research, but in the research of many neuroscientists and neurologists today, is the need to image layers of the cerebral cortex, so that the cerebral cortex is not imaged in PET anymore as a band or blurred, but as multiple layers. Maybe not all of them, as you know, there are 6 of them, but at least at 2 or 3 of those. And the reason, because we have in PET neuroreceptors that map many of the functions we're looking at. And there are different types of receptors in those different layers. So for example, if you look at autoradiography, so with PET imagers that are looked at in brain slices here, you can see that the muscarinic and the noradrenergic alpha receptors are expressed differently in the occipital cortex versus the motor sensory cortex. And if we go back 1 minute again to aging and Alzheimer's disease, we and others have shown that the tau progression occurs in a set of stages, and the cortical deposition is lamina specific, meaning that very early stages of BRAC you would see deposition in some of the layers, and layers like layer 5 is not seen until much later. The challenge is this bar of 2 millimeter, I hope you can see here, that we would like to break. We have to be at least at 1 millimeter in order to be able to quantify something of this size. So that's another area where this high resolution is important, and by no means these are the only ones. So where have we been in terms of PET in the field? If you look at all the systems that are available today, you'll see that most of them, and even when you're subtracting non-collinearity, which is something that is affecting resolution, so the resolution would be even worse than that, we are far from where we would like to be with the SAVANT system, which is here. What we're building is going to be around 1.1, 1.05 millimeter, will be far below everything else that is available today, even those systems that have 101 crystal photosensor, but they're used in small animal imaging. These are small animal imaging, and these are the clinical scanners. So the way we accomplish this is by having one-to-one coding in between the crystal scintillator arrays and the avalanche photodiodes that we'll be using. That one-to-one coupling means there's no coding effect, there is no code sharing, there's no crystal sharing of light, and therefore, or block detectors that are sharing light, and therefore there's no error in the positioning of where those events are. And that means we have a very high intrinsic resolution, but also high count rate. And because these APDs and silicon PMs are insensitive to the magnetic field, the system is going to be very compact. So we'll be able to integrate that into an MR system in the future. The cost for this is going to be first in terms of ASICs, because to get a compact system, you need to have new ASICs or new electronics to be developed. Thermal load, what we've talked about before, has been in small animal imaging. We're talking about a whole brain scanner here, and then reducing cost. So here's the system that we have developed and that we are building. The SAVANT system models the depth of interaction as well. So in fact, we have 2 layers of crystal instead of 1, a fast LGSO and a slow LGSO. And it has an opening of about 40 centimeters. So that's plenty space for brain, for fitting a patient brain with 23 centimeter of axial field in length. So no risk of truncation here. But that comes at the price of 258,000 crystals. We have more crystals here than in a whole body scanner that would be an opening of 75 or 80 centimeters. And the reason is because the crystal is many-fold smaller than what you would have in a regular system. In fact, the crystal is, you can think of it as a needle that is 1 by 1 millimeter, and that is 2 centimeters in length. And here's where we are in terms of progress on the construction of the scanner. The hardest part in terms of the electronics is done. I say it's the hardest part because those have to be redesigned from scratch, and there are very few sites in the world that are able to do the electronics design and motherboards and ASICs, and that has been finished. I'm happy to report we're done with that. The LISMO DAC is done. The ancillary systems are also done. Cooling, which was one of the big challenges in terms of heat dissipation for such a long axial field with APDs, has been done, and it's very stable. And I'll show also some results on the motion tracking and software that goes post instrumentation. We are still in the process of buying crystals, cutting crystals, and polishing crystals. And here's the performance in terms of spatial resolution that we talked about. So if you look at, if we didn't use no depth of interaction, you'd see that as you go away from the center, say, let's look at 4 centimeters here, you're going to have a spatial resolution that is going to start deteriorating as you go away from the center. And that's problematic because for the cortex, for example, if you want to look at the cortical layers, we need to be with a good resolution throughout the field. With depth of interaction, we have that. We're at 1.2. And sensitivity is the same, about 6%. If we look now at kind of the performance with a hotspot phantom, this is the SAVANT system, and you can see that we have 1 millimeter resolution. You can see all the rods at 1 millimeter. Compare that to the HRRT, which is the best brain scanner available today. There are 17 of those, and they are not serviced by the manufacturer anymore. So those systems are at around 2.4 millimeters. And the biograph vision is another point of comparison, because that's the whole body system that is available today, with one of the better spatial resolutions clinically, and we're at 3 millimeters. And note that the counts are half those of the HRT or the biograph with our system, but note that the image quality is far superior. And the reason for that is from information signal theory. All events are detected in the correct exact location, so you're not losing in terms of information theory by having blurring that you have to recover by correction afterwards. So image quality is still much better, even though you have fewer counts. And when you go off center, and this is an extreme case, we're going at 10 centimeter off center, but you see that the deterioration in terms of spatial resolution if you don't have depth of interaction. And with the SAVANT, we're still at 1.2. With the biograph vision of the HRT, there we're at far worse, with a 3 millimeter or 4 millimeter. And here's now images from the histology-based brain phantom. This is, note the voxel dimension is 0.4 millimeter. Until very recently, we always used the Zubal phantom because that was plenty for imaging with PET. It's a digitized MR, and that was fine because at 1.2 millimeter pixel size, this is more than you need with a 4 millimeter resolution. But when you go to 1 millimeter resolution, you can see that you need a very small, literally histology-based phantoms to look at what your image resolution is. And I hope that you can see in those images, they're starting to look more like an MR than what we're used to in PET. If we look at one slide, maybe, you can see here's the internal cortex that we're looking for, and you can see that it's very well delineated, as well as the precuneus. And if you compare those to the two systems we described, the HRT dedicated brain and the biograph, you can see the exquisite improvement in image resolution, not only in the cortex, but also in the deep structures equally well, because with depth of interaction, we have uniform resolution about 1.1 millimeter across the field compared to the HRT or to the biograph vision. So have we reached the limit yet? Are we there? And the answer is no. There's still improvement we can do. What we're doing today is no time of flight with the scanner because it's an APD system, and that means it's a classical reconstruction. As we go to clinical scanners in the 250 picosecond, you're not going to improve resolution, but you will have gain in terms of signal-to-noise ratio because you're reducing the line of response where you're reconstructing to the line of detection. We're aiming, and I'll show in the next slide, where we're going with 40 picosecond timing resolution. Where there we think there will be less need for reconstruction, and we will and you'll start getting a gain in spatial resolution, because as you get to the 10 picosecond, you will not need reconstruction, and you will have some improvement in image spatial resolution. And note that the positron range, I'm often asked this, in water for fluorine 18 is 0.1 millimeter. So we're far from that. That is not yet a big limitation for spatial resolution. So how are we going to do this? We're working on with the European Consortium on building a hybrid sensor that would have a 3D integration, that would allow for silicon photomultipliers and for the ultra-fast electronics to be integrated together with a CMOS process. We're aiming for about 50 picosecond to start. This is scalable. The architecture is 3D, and it allows different arbitrary large arrays, so you can easily imagine this becoming a total body for other cancer applications as well as in the brain. And it's low cost. Once the electronics are designed, that's the expensive part. The scaling of those with mass production goes very, very quickly down. Just an appetizer, here's what you would have with 4 of those detectors, just four, as compared to a whole brain scanner. If you had a whole body scanner that is clinically available, you can see the image quality very similar. The last piece I would like to leave you with is the work we're doing on the post-processing side. Never underestimate the power of what you can do after the images are done. So that's not instrumentation, that's more image analysis. And the first is denoising. And the reason why denoising is important is because, we are detecting the fewer counts. So we would like to be able to, although they're all perfectly well located, we'd like to have denoising because we are imaging dynamically. We're going to image in short frames. The image I showed you are in the 4-6 minute kind of acquisitions would like to be in the 1-2 minute for some of the, or 30 seconds for some of the dynamic imaging we're doing with tau, for example. So there, denoising is important. And what we're doing is, instead of using the noise as an input along with corrupted image, which has been what other people have reported on, we're using the MR as the input along with the corrupted image, and this is the kind of results you can get. I'm showing you here results from denoised FDG scans of glycolytic uptake in the brain, and you can appreciate the improvement in denoising. The second piece we're doing is in terms of spatial resolution. Here we are sampling very, very fast the brain temporarily with an optical device, much faster than motion is happening. So we can correct for motion, but we also oversample below the pixel size the image so that we can recover more resolution. That's what we call super resolution. And here's the kind of images you could get. This is your conventional PET when you're just correcting for motion. This is when you're doing super resolution compared to the reference. And you can see the improvement in image resolution and image contrast you can get. And finally, the last piece is, yes, motion is essential when you are correct, when you want to have very high spatial resolution. But we also still need to correct for partial volume, because when we talk about cortical layers or locus ceruleus, we're in the millimeters. And here we are using the patch similarity between the PET and MR to look at recovering partial volume within the reconstruction formalism. So we add the kernel within our projector, and you can see that we obtain very impressive images here in terms of cortical ribbon recovery in glucose, and in the tau images we talked about when we started. We come back to that. Again, you have a much better improvement, much greater delineation of the tau deposition, which is very important in terms of staging in the cortex as well as in the neocortex and in the internal cortex. With that, I would like to thank again the organizers for inviting us to give this talk. I would like to acknowledge our teams, the team at the Gordon Center at Mass General Hospital and Harvard Medical School, and the team in Sherbrooke, the University of Sherbrooke in Quebec, and of course our program directors, Dr. Shumin Wang and George Zubal, who keep us on our toes to try to get things done on time. And with that, thank you for your attention, and I will take any questions you may have in the chat period.